To this morning's session, which I'm very pleased and happy to point out is going to have a lot of talks on P53. And we're going to start off, indeed, with the father of P53, maybe the grandfather of P53, Arnold Levine, who, as always, has a fascinating story to tell. So.
Good morning, children. The work I'm going to tell you about is done by two postdocs now, one assistant professor. Chrissy Dungeon is an experimentalist who works in the lab and has done all the experimental work, and Chang Chan is a theorist and or bioinformaticist and has done all the informatics work that I'm going to tell you about. He's now an assistant professor at the Cancer Institute in New Jersey. This is a slide that is actually on the P53 website. Thanks. I have to figure out how this actually works, if I can get. Well, I've got it oriented well, so maybe I can now figure out. Oh, okay, I got it, yeah. P53 website, and thanks to Magali Oliveira, who is the person who curates the P53 website, and it's a summary of a great experiment going on around the world today at, obviously from the TCGA experiments that have been done, funded by NIH all over the country, and the Broad and Wellcome Trust and Beijing Genome Center. And this is probably 6 months to 9 months old now, or in the rears now. And that is the unbiased sequencing of tumors and the unbiased sequencing of peripheral blood cells, and looking at the differences and plotting mutations that are found in 125 of the most frequently mutated genes, tumor suppressor genes and oncogenes in this case, all of which are validated by mouse models and 25 years worth of research. And in this three-dimensional plot, there's 36 tumors, which it's not meant that you're supposed to read any of this, but it just gives an overall impression. 36 tumors that are the tumor types, the 125 most frequently mutated cancer genes, and then the frequency from 0 through 100%. And the take-home message of the skyscraper plot, nice way to put it, is that roughly, and it is rough, that you can put these genes into about 3 classes. There are some oncogenes and tumor suppressor genes that are kind of 1 to 5%ers. That means in all the tumors that have been looked at, the 36 types of tumors, they represent the 1 to 5%. In some cases, they are very high in a particular tumor. So I think BRAF is a very good example of that. It's quite high in melanoma, maybe up to 50%, but it's maybe 6% or 7% in colon cancer and a number of other cancers. These are actually, oddly enough, have been some of the easiest oncogenes to drug, and the great majority of drugs that have been made by the pharmaceutical industry are in this 1 to 5% range. Which is a bit of a disappointment in some sense. There's a next class, are I'd say 30 to 50%ers, and they represent PI3 kinase, and it goes a little higher if you put in the whole pathway of PI3 kinase. That's an example. I used to say the 30 to 50%ers are undrugged, but actually the PI3 kinase delta drug has just been approved for B-cell lymphomas 2 weeks ago. So that's the beginning of an alpha, beta, gamma set of inhibitors in the PI3 kinase. But there are other examples there, RAS, MYC, we heard about, which are all very high, frequently BCL2, which give rise to anti-apoptotic phenomenon. They're in this 30% range, to 50% range, and there are no drugs. There are some drugs in the clinic that are moving in that direction, but there's only one single drug approved for the delta PI3 kinase. And then, unsurprising, because this shows up at the P53 website, is this line for P53. And there, about 55 to 60% of all tumors have mutations in the p53 gene. And if you then start to add up other ways to defeat the function or to modulate down the function of p53 in tumor cells, it can go up to about 70% of tumors that you could characterize as having compromised p53 function. Now, there are a lot of variation in this. For example, in ovarian cancers, I think literally all of the serous ovarian cancers, literally 100% have p53 mutations. But in other tumors like teratocarcinomas, testicular teratocarcinomas, which really found frequently in younger males, for example, it's 1 to 2% p53 mutations. Then there are also variations, because to know how to count is an interesting problem. There are variations in that if you're diagnosed with prostate cancer, for example, the frequency of p53 mutations is about 3 or 4%. It's not very high. But after the anti-androgen therapy fails, which it almost inevitably does, after anti-androgen therapy fails, the frequency of p53 mutations is up about 45 or 50% in pancreatic cancers. So it depends when you measure it, and certainly, and you'll see why in a minute, certainly a reasonable percentage of these mutations can arise because of the therapy themselves and have actually adverse consequences because of the therapy. Now that we have sequence information and it's unbiased, we have to learn to start to deal with that therapy has an inadvertent set of effects of making tumors worse as well as giving rise to a certain percentage of cures. But the take-home message of this slide is that in a great majority of human cancers, p53 mutations happen, and the question is why? If we really understood the functions of p53 and were able to replace them in some way, either through synthetic lethality or other mechanisms, we would have made a very big impact on human cancers. And so the question becomes, what are the functions of p53? And the next slide actually just is a little out-of-date brief review of the functions of p53. I think everybody in the field would agree that p53 is a responder to a wide variety of stresses. In fact, a surprisingly wide variety of stresses, which are found in a list up here. Most commonly DNA damage, and that's probably the most common stress that activates p53. When I say activate, I mean it takes a low level of a latent form of a transcription factor, brings the level of the factor up, modifies the protein, and starts to roll in a program of transcription, which is p53's response to stress. But hypoxia, nutrient deprivation, and ribonucleoside triphosphate levels, so a link to metabolic sensors is important in p53 stresses. Spindle damage is sensed, heat and cold shock, a whole variety of telomere length erosion, whole variety of responses, and even oncogene activation. So ETS and beta-catenin and MYC, so RAS activation, for example, is sensed. So this is a molecule that's sensing signal transduction pathway abnormalities. And in sensing these signal transduction abnormalities, it itself gets activated, activated meaning high concentrations, protein modifications, and transcriptional responses. Almost all of the signaling from stress to p53 goes through the ubiquitin ligase for p53, MDM2. It either increases its activity, in which case p53 levels go down. That happens when the cell is committed to cell division, for example. AKT is a kinase that phosphorylates MDM2 and will increase the activity of MDM2, decrease the activity of p53. So in that sense, it moves away from the commitment to cell division, p53, and surveillance is paralyzed. On the other hand, something like DNA damage working through the ATM pathway, in fact, inactivates MDM2, which then inhibits p53, and all of p53 regulation is at the post-translational level in its half-life. So it goes from as short in T cells, which I'll talk about, as short as 6 minutes half-life, up very rapidly from 6 minutes to 12 minutes to 24 minutes to 48 minutes. And the concentration rises rapidly within an hour, for example. The protein modifications occur, and there are a lot of them. And then a transcriptional program is rolled out. The first of the three common responses have been apoptosis. There's a program for freeing NOx and PUMA, free cytochrome C from the mitochondria. That works with APAF1 through the Cas-based pathway in a classical apoptotic response. It is inhibited by BCL2. Senescence, which also calls in an innate immune response. And so there's an immunological response as well as a senescent cell. Cell cycle arrest, which is in some cases reversible. But there also are other responses that are not on here, metabolic responses. It changes cells back from a preferentially Warburg response, that is to say use of high levels of glucose and aerobic glycolysis, lactate secretion, lactic acid secretion, to an oxidative phosphorylation response. Glutathione levels rise to protect the cell. For example, NADPH levels rise to protect the cell. So that P53 is really running a surveillance survival mechanism as well as a death mechanism if the stresses are too great. Now, that would be, all of this would be absolutely perfect explanation for what a tumor suppressor is working, except for the fact that as people have teased this apart, things seem to fall by the wayside. For example, the major genes for tumor suppression for apoptosis appear to be NOXA and PUMA, which are P53 transcriptional targets. They activate cytochrome C release. They activate the apoptotic pathway. But in the mouse, you can knock out NOXA, you can knock out PUMA, you can knock out both, and it doesn't change the ability of P53 to surveil against tumors. In other words, its tumor suppression is not totally dependent upon apoptosis. It may be necessary, but it's certainly not sufficient, right? And that's true for senescence. That's true for cell cycle arrest. P21 is a major cell cycle arrest, an inhibitor of cyclin E, CDK2. Yet you knock out P21, and it doesn't take the mouse to pro-tumorigenesis, right? And so there's been a rather systematic way of looking for the mechanism of tumor suppression, or mechanisms, probably plural, right? And we've yet to really uncover it in a clear sense, right? So given that as the problem, we decided to just take a step back. It's back because this mouse I'm going to use in the model that I'm going to talk about is very old. It's a mouse made first by Tyler Jacks and Larry Donauer. And this is the P53 knockout mouse. And this will just show you what happens in a P53 knockout mouse. Because when you don't know the mechanism of suppression, it might be good to look at the nature of the tumor in some detail that forms. And if you can look at it, you'll find something that has gone wrong in the absence of P53 that maybe suppression acts upon, right? So this is the age of the mouse. The P53 knockout mouse is shown here, this homozygous knockout mouse. Survival, and you can see that the mouse dies at a fairly young age, certainly by 6 months or so. The great majority of mice have succumbed to tumors, and their survival is compromised. The heterozygote is in the middle, and the wild type is here. So there's a terrific difference. P53, this is the ultimate test for its tumor suppressor activity. The nature of the tumors is, in the case of the homozygous knockout mouse, 65% T-cell lymphomas. These are thymic lymphomas. They're CD4, CD8 double positive thymic lymphomas, and I'll talk about them in some detail. But there are also soft tissue sarcomas and other kinds of hard tissue sarcomas. And in the heterozygote, which is more like the condition that some humans have of inheriting one allele, mutant allele, there are a lot of sarcomas, and although thymic lymphomas are not found in humans, the sarcomas are found in humans, adrenal corticocarcinomas, breast cancers, some brain tumors, some lymphomas are found in humans. So this is the model system I'd like to look at, and I'd like to be able to tear it apart and ask certain questions. So let me just phrase some of the questions. In a P53 knockout mouse, what is the probability of getting a tumor, a transformation event, right? Does it happen once? Does it happen once a week? Does it happen once a day? And are the tumors multiclonal, and what is the heterogeneity of the multiclonal tumors? Do those multiclonal tumors or oligoclonal tumors compete with each other? And is there a winner eventually? And what does that winner look like with regard to oncogenes and tumor suppressor gene mutations that are occurring in the absence of p53? And what is the nature of the mutations that are occurring in the absence of p53? Now, none of this actually, oddly enough, even though the mouse has been available for 10 or more years, none of this has really been investigated. And it turned out thymic lymphomas allow you to ask some of the questions I've just talked about. And the reason is in retrospect obvious, but I don't think anybody's sort of done it yet. And that is that as each clone arises in the thymus, it gets a unique zip code on the cell surface, and that is its T cell receptor. The VDJ joining region, and also the new nucleotides that are put in the VDJ region by terminal transferase, which adds nucleotides, gives you a unique signature for a clone. And the number of reads of DNA of that clone gives you the number of T cells that are in the thymus if you sequence very deep, right? So just by doing DNA sequencing on those tumors as a function of time, one can ask, are there multiple tumors? And you can declare a transformation event or a tumorigenic event as being a statistically significant increase over the background of the average frequency of clones in the thymus, in the wild-type thymus, right? So it's a pretty straightforward analysis, and it's all dependent on sequencing. So let me take you through this. We heard a little lesson about B-cell development last night, I think, and so this is a little lesson on T-cell development under the assumption that we've been a long time since some of us have taken immunology, right? So clearly this all starts with the hematopoietic cell, the CD34 cell in the bone marrow. It divides several times and makes a precursor of the lymphoid cells, divides again, and the precursors of T cells move into the thymus. And this is all during embryonic life. And the first cell to show up in the thymus has these kind of markers on it, and VDJ joining and recombination begins in what's called a double negative number 1 cell. And then some of the markers change, and you continue recombination of the DJ region, for example, in a double negative 2 cells. You continue VDJ joining here in a triple negative. During embryonic life, for instance, the gamma delta chains, which form gamma delta T cells on the surface, are formed first. They're the T cells that are in your skin predominantly. And the alpha betas are then formed late in embryonic life, early in life after birth, for example. And then for the first time, the beta receptor is inserted in a double negative 4 cell, and a surrogate for the alpha receptor is put in for it. And this results, the fact that you've got a successful VDJ joining and the terminal transferase reaction, it makes a protein, it inserts into the membrane. This results in a terrific proliferation of the clones that form. And then the clones are tested against the HLA type, or the H2 type in this particular case, class 1 antigens, class 2 antigens. And when tested, there's selection against certain self clones. And then finally, a double positive cell arises, which is CD4, CD8 positive, CD3 positive, which is indicative of a T cell. It has an alpha receptor and a beta receptor, and that's the cell that actually accumulates in a thymic lymphoma. So this is the cell we will get in the thymus. It's blocked for further differentiation into CD4 and CD8 cells, and so this mouse is actually immunocompromised but dies of its tumors early in life. We have sequenced in each case just the beta receptor. When we've looked at the alpha receptor, nothing really changes. So any zip code that's useful to look at clones or clone numbers will sequence through the beta receptor. Okay? All right. So let me give you an example of what happens under normal circumstances, because we have to know the number of clones that appear in normal mice to give that background level above which DNA reads are giving rise to thymic lymphomas. So this is the thymus, and these are just the top 10 clones in their frequency in the thymus. The highest clone is 0.03%, so it's very low. And then there are examples of a 0.02, and there's examples of a 0.18. Now, this is all from DNA reads, and their frequencies give us, we know there are, at birth, about 10 of the 8th cells in the thymus, the T cells, and then they go to 10 to the 9th. So we know the total number of cells, we know our reads. We're not deep sequencing very deep because we get to statistics that are good enough very early. But this gives us a percentage, and that's why we stay in percentages. I'll give you some raw numbers in a few minutes. And so there's a very low level of clones. There are 138,582 unique sequences in the thymus. So the thymus is a place for a lot of unique sequences that are sitting around waiting to be called forth if needed. This is what happens in the spleen, if just looking at T cells again. In the spleen, now the frequency is up about tenfold. There's 0.22s and 0.21s, and you see the frequency there, 5,913 unique clones. So this moved out into the periphery and counted antigens. There's a tenfold increase in the T cell clones, and there are very much fewer unique clones. And then if you just look at peripheral blood, there are 21,000 unique clones. There's a 3%, a 3.5%, and on. This was our first tip that maybe our mouse colony had a pathogen in it. Actually, it's actually an excellent surveillance mechanism for, this was not good to report to the mouse colony people, but this was a first tip that there was something dominating the environment of our mouse colony. Maybe it was the investigator for all I know, but whatever the reason is that having that kind of level is actually not unusual. And it's sort of a nice way to do surveillance if you don't mind paying the bills for Illumina sequencing. Right? Okay, so this is the background rates. And now let's take a look at what happens as a function of time, right? Because we don't know yet when the thymic lymphoma shows up. It may show up in the embryo, it may show up at 5 weeks or whatever, because P53, missing P53 knockout is not sufficient for the tumors. It may be necessary, but it's not sufficient. Okay? So here's an example of the unique clones. This is just unique clone numbers from a moderately DNA sequencing. This is embryonic 17. This will be the time elements we've used throughout. Embryonic 17, 3 weeks, 6 weeks, 9 weeks, 20 weeks. And this is a frank tumor, as you'll see. We did males and females because the immune system is sexually dimorphic. And because of that, we worried that the tumors may be sexually dimorphic and we may be getting two kinds of responses in tumor rates, but that's not true. Everything seems to be the same by this analysis in the thymic lymphomas. So we did 2 males and 2 females. This is the wild type. They're about, if you sequence this deep, it's an arbitrary depth in sequencing. They're about somewhere between 31,000 and 44,000, and that's the variance around it, unique T cells. And the knockout mouse is about the same. Then at 3 weeks, there are a male and 2 females. This is, the numbers have gone up slightly here, but they're in the same range. This is statistically not significant. If you double the number of reads, you increase the numbers here. By 6 weeks, it's about the same, 49 to 56,000 unique clones, about the same at 6 weeks. So there's just no evidence from unique clones that anything is happening. At 9 weeks, something does happen that's statistically significant, between 33 and 49,000 in these 3 mice, and 20 to 27,000 in these mice. By 20 weeks, if you just look at the females, and here's males and females, this is down to from 20,000 to 5,000, 3,000, 4,000 unique clones. Now, the reason this is happening by looking at unique clones is that there is, in fact, a tumor arising, and it's replicating like hell, and the number of T cells in the thymus stays roughly constant, so it's squeezing out all the unique clones. So we see loss of unique clones as the first indication. Now I'm going to show you the top 2 clones in terms of numbers, which is the reciprocal of the unique clones. Okay? All right, so here's E17, the wild type. This is the percentage of the 0.16, 0.17%. This is not the absolute numbers again, just to give you a different way to see it. This is the first clone, this is the second clone. So there are multiple clones, obviously, and this is the most common ones, and there's no difference between the knockout and the wild type. 3 weeks, there's no difference. 6 weeks, there's no difference. By 9 weeks, 0.08, 0.07, 0.16, 2.9, 1.2, 21%, 13%. Now we have clones that are arising, and these are just the top two. You'll see in a minute, they go deep into 10 different clones that have arisen. So we can say rather clearly a couple of things right away. This tumor is oligoclonal. It has many clones that are arising that have different T-cell receptors, and they coexist at this period of 9 weeks. And 9 weeks is the first time we statistically significantly see reads that are above the background level. What's nice is by 20 weeks, this stays about the same, but now look what happened. A 70%er and a 28%er, 98% of the tumor is 2 clones, right? So that just shows there are winners, right? It's oligoclonal at 10 of them, 15 of them throughout this early period, and then they narrow down to 2 clones, which are winners. So we know that there are subsequent mutations occurring that I'll talk about, and you'll see that there's both an order of mutations being selected, and you'll see that the same mutations are being selected for in this pathway, right? Yeah, but not all. Here's one that's 43%, 19%, 70%, 5%. This is 75%, right? And so forth. So a good way to show that is here are the 4 mice. So while this is 70 and 28, that's 98%. There's a background rate after that. This is all background rate. This is all, by the way, the knockout mouse at that 20-week period. These are the winners, right? So here's a 43, an 18, a 5.7, a 3.7, a 3.4, a 2. So there are a lot of new clones arising all the time, constantly challenging. It's possible, of course, since we know the time and the number of clones, I'll estimate frequencies for you of mutation frequencies. I'll estimate clonal frequencies in a few minutes. But this gives a lot of detailed information about the origin of the tumor and the evolution of the tumor in the absence of p53. 14%, right? 8%, 1%, and so forth. So you can see that there are dominant clones arising, but that they are of varying differences in mice. So the mice are genetically identical. They all have p53 mutations, but they have variations, stochastic variations throughout. Okay, so then we sequenced exons, and we did this in first 3 mice and then more. And I'm just going to show you some of the changes that have occurred. It turns out that the point mutation frequency is not exceptionally high, and I'll compare that in a few minutes. But this is actually the genomic rearrangements that we see from the sequencing experiment. Now, so here's the 20 chromosomes of the mouse. This happens to be a male, so that the X chromosome only occurs in 1 copy. So this is the level at which it would be if it occurred in 2 copies and 1 copy, right? And one of the things that you see, and if it's red, it's amplified or aneuploid. In this case, these are aneuploidies. The aneuploidies are very common in these tumors. And that was well known. P53 surveils against aneuploidies. The absence of P53 results in high aneuploidian tumors. And this confirms that. This is a really great example of something seen in human tumors that don't have P53 called chromatripsis. It is a series of thought to be brought about by recombination events of amplifications and deletions on the same chromosome. So here are deletions in green and amplifications in red on the same chromosome. Right? And for example, chromosome 5 in every tumor we've looked at is always aneuploid. Chromosome 3 has a lot of chromatripsis on it. So that patterns start to arise, and I think you'll hear from Luke Hantley some very similar kinds of observations and conclusions. Patterns seem to arise in the genomic instability, which look like they're reproducible from tumor to tumor to tumor, although in our case, not as reproducible as you'll see in Lou's case. Right? So the take-home message here is there is an awful lot of genomic instability across the genome of these mice. All right. Now, I promised you some numbers, so let me give you some numbers. The frequency of point mutations is about 1 per megabase. Now, that's not remarkable, and in fact, it's not even the highest numbers one sees. In melanoma, you'll see about 10 point mutations per megabase. In breast, in pancreatic, in ovarian, you'll see about 1 mutation per megabase. So this is comparable to data that one sees in humans for point mutations. It's high, obviously, by every standard. It's 1,000-fold higher than it is the germline mutation rate, but it is not a remarkably high mutation frequency. This is a frequency, not a rate. And it is not a remarkable higher frequency than what one sees in human tumors, so that's not a surprise. However, the number of copy number variations is very large. In the tumors we've sequenced, it's between 276 and 422 per genome. And this shows a lot of genomic instability in the tumor. And as you'll see in a few minutes, every oncogene and tumor suppressor gene that we've lost is due to, every tumor suppressor gene we've lost due to deletion. Every oncogene is due to amplification, right? And so this is driving the tumor drivers to really, the nature of the mutations are through these copy number variations. And then a particularly interesting number with a large variance is the number of transformed clones that arise between 9 weeks, which was the time we first saw 2%ers, all the way to 20 weeks when we see winners, is somewhere between 1 transformation event per week and 1 transformation event per day. So there's a lot of stochastic aspects to this. There must be other things happening in the mutation. Okay, I'm sorry. I'll go fast now. I can move fast. All right, so let me just compare that for you. This is from the Broad Institute. It's point mutation frequencies. The one that I showed you is here. It's in the neighborhood of breast, pancreatic, and so forth. The melanomas here. I'm just going to move fast. And I'll finish with the mutation frequencies we see. Right. This is an interesting thing because one of the mutations we saw was a deletion of Pten gene. Here's the sequence from 3 mice. They're different mice. There was a deletion of 88% of the reads found across here in the mouse, of 97% of the reads across here, and 85 here. That means 3 different mice, 3 different deletions. However, every single clone in that mouse, even though they were oligoclonal and different clones, had the same deletion. Right? So that's a surprise. How do you have the same deletion in a mouse in all different clones, and yet different ones between mice? Well, the answer is it arises before, or it's selected for, before the T-cell receptor is inserted, right? And so all the clones get it, right? So this is the earliest mutation we see before T-cell receptor is put in, is Pten. And finally, this just shows all of the mutations that we see. This is the early 6 weeks. Here's the Pten levels in both knockout and wild type. This is prior to any tumors, so you get plenty of Pten. Here's the deletions you see. There are 3 mice that are heterozygous for Pten, and the rest have the deletions for Pten. There's a CDK6 amplification in the tumors, right, which is not seen in the normal, and cyclin D1, D2, and D3. D3 is a T-cell cyclin, yet all of the different cyclins are amplified in different tumors in different ways that run CDK6. So the first is a metabolic change, Pten. The second is a cell cycle change. RB is lost, which is a redundancy for this CDK6. RB is lost in 3 tumors, right? And then Ikaros, which is a differentiation factor in the Notch pathway. Ikaros is actually making splice variants, which are negative regulators, right? Which prevents differentiation into CD8 and CD4 cells. And it actually, what Ikaros does under normal conditions is turn off a gene called HES, and you see HES1 is on in many tumors, but there's some heterogeneity here. So what's quite surprising is that the order of the mutations and the nature of the mutations is the same in many independent tumors, once you start with a gatekeeper P53-, right? And so in that way, we conclude that there's a pathway here that is the simplest pathway to go through, that gives rise to it, and that the major defect is genomic instability, which is what P53 is surveilling. Thank you. I'll stop here. Sorry. I thought I had so much time. Okay. Okay. I'm on time. Okay, good. It's flashing red. Yeah, yeah. I knew at Cold Spring Harbor, flashing red is not good.
So we actually have 10 minutes for questions.
Oh, well, okay. I have 30 more slides if you want.
Would you rather have questions or 30 more slides?
Yes. I was teasing.
So you showed that the oligoclonal tumors, in a lot of cases, like all of them had lost Pten, and so it was obviously an early event. In the ones that the mice up there where you don't have it lost, at least at a whole population level, if you were to look at the different clones, would you find that some of them had lost them and some of them hadn't? So then the order wouldn't then have been an early event in that tumor?
So clearly, this kind of heterogeneity says it's happened after the T-cell receptors between them. And exactly when that arises and when Icaros arises, we don't know. I'm kind of guessing that the next driver in line is cell cycle, and then a differentiation block gives you a piling up. What's very nice is each of these mutations are in pathways, metabolic pathway, and then cell cycle pathway, and then differentiation pathway that makes sense. And it's these very pathways. For instance, Notch is probably the most common mutation in human T-cell lymphomas, and Icaros is in that pathway. So not using the same genes, but it's using, in the pathway sense, a very similar set of genes to the T-cell lymphomas in humans. Yes?
Arnie, so I guess there are two general mechanisms. One is selection for convergent evolution.
Yeah.
And the other is that there are structural reasons why certain mutations tend to arise.
Right.
So obviously, I don't think anybody's ever done it, but if you were to do this sort of analysis on an individual non-transformed thymocyte,
Yes.
how much diversity do you think that there would be?
So we're just getting, that's a very good question. What happens in a thymic lymphoma that arises with its origin in the presence of P53, right? And of course, that's this experiment where your mouse colony goes to infinity, and you look for the rare though thymic lymphomas do happen in this background, that's Black 6 background. I've been very fortunate to been able to collect a bunch of thymic lymphomas that come from other labs that are more spontaneous. As a generalization, there's a bit of literature here on it, and they tend to be driven, oddly enough, by retrovirus movement, right? And that's a very interesting possibility because genomic instability is, of course, like retrovirus movement, or it's like the insertion element movements we've heard about last night and so forth. So there's some common features that could be driving it, but we don't know yet because we haven't sequenced that yet. But it's a very useful comparison, the question you're asking. Would it change the subsequent mutations in the selections? I don't know. And I do think there is a structural issue here. If you have genomic instability as your driver from the beginning, you are going to select for deletions and amplifications, right? And anything that had to be a point mutation couldn't be anything else, right? Would tend to be pushed into the background. So there is it's the genomic driver that actually changes the nature of the mutations that you might select, yeah.
It's a great talk. I'm curious about the distribution of tumor in a knockout. I mean, 65% lymphoma I kind of expected because of high proliferation and the checkpoint protein is very important. But why osteosarcoma and soft tissue sarcoma? Is there a rationale behind it?
So you're asking a question which I think is a sort of very general question. Why is it that different tumor suppressor genes tend to have tissue specificity when they're inherited, tend to have tissue specificity when they're inherited and give rise to tumors? So Lymphoma patients, the very first tumor, as a function of age now, I'm going to go, the very first tumor you see in Lymphoma patients, not in all, but some, that's the heterozygote P53, are adrenocortical carcinomas. Then as you get older, you start to see some sarcomas. For instance, in teenage years, you see osteogenic sarcoma, right? From 20 to 45, you'll see breast tumors. Right? And you'll see leiomyosarcomas at early age as well. There's an actually very interesting parallel, that's all it is, association, between the age of onset of different tumors and the fact that that tissue is actively replicating as a function of age, right? Breast in the 20 to 40 range, leiomyosarcomas and osteosarcomas in the more teenage years, adrenal corticocarcinomas in young. So there may be something to do with these tumor suppression surveillance mechanisms of actively dividing precursors. It could be stem cell-like issues, it could be division issues, it could be building the organism issues, but that's what happens for early onset Li-Fraumeni syndrome. Why they are sarcomas, I mean, the major tumor is a sarcoma, and we don't have the slightest idea of tissue specificity, although I've given you a correlation for that.
A question in the back?
So when you compare your 9-week-old to your 20-week-old populations, have you looked at which loss of which genes determines whether the losers lose?
Yeah. So I think we're in a little bit of a situation a lot like, yeah, so that you're asking a question we had hoped I could get up, or I had hoped anyhow, I could get up and answer. I can't, and you'll see why. It'd be very interesting to know what is acting subsequent to everything to give you an optimal tumor, right? Because maybe you'll pick up metabolic processes, a whole bunch of interesting things. But we're in the position that the Sleeping Beauty issue that we heard about last night, there's so many insertions, there's so many rearrangements, there's so many point mutations across the genome that I can make a list, but it isn't explicable to me what it is that is giving rise to that selection. And it doesn't pay to guess from the list by putting them in one at a time as well. So this hasn't, the place where this whole approach has been weakened is to really get at the question you're asking. What is it that gives you a winner, right? If all of them have about the same maintenance, to get you there, why are you a 70%er and a 28%er who looks pretty much the same, right? And I don't really have a good answer for that yet.
Have you ever done similar experiments with mice that have mutant P53 instead of no P53?
Yes, we have. So the mutant P53, among its properties, is that it has it often has a gain of function, and it's that in Gigi Lozano's work, the gain of function has seen itself as increased metastasis in mice, for example. And we have done this with the 172 mouse, but homozygous 172 mouse, and get exactly the same results. So it isn't obvious what the gain of function is for the lymphoma, but maybe a lymphoma isn't the best tumor to be looking for that gain of function if migration or if other kinds of mobility and expansion is the issue. Maybe we're not looking in the right place yet.
So you showed this amazing diversity of the early event, the Pten loss, and I assume you know that the one that's still there is also somewhat functional, or do you?
No, so one of the three had a point mutation, and I don't know for sure. I know there's a protein there, but I don't know for sure it's functional.
Okay.
And so there were three that were haploid, essentially, for that Pten. One of them is out because it has a point mutation that it pretty clearly knocks it out. The other two I can't really say. Whether haploinsufficiency is an issue or whatever is the issue.
And there's no, so they don't show alterations that stand out that are particular for those clones that
I'll give you an explanation which it seems to me to be reasonable explanation. There are many kinds of mutations that occur during development. The one that's being picked up in the T cell compartment is Pten. Why? Because that gives you the ability to use glucose at a high rate and make you grow like hell, right? So suddenly the Pten it's not that it occurs selectively, it is selected for because of its growth. Now it will form 5 tumors, right, in a mouse with the same Pten because its clonal size has increased at that time. We've begun to look at other parts of the hematopoietic lineage to ask how early this really occurs, right? And it looks like it occurs mainly in the early T cell lineage. It isn't seen in the B cell lineage, for example, which would be the closest one for divergence. So, but once happened, it selected very quickly to take over the thymus, if you will. So my guess is if I had, even though you were haploid for it and it was there, it's probably being selected in some way because it's so homogeneous. Yeah.
Okay, last question.
Stefan Pfister's lab, in studying medulloblastoma, has identified a very high rate of chromothripsis. Essentially, all of it's associated with Leishmania, and the vast majority of those patients, Leishmania was diagnosed in the child and then realized in the family, so there's no family history.
Right.
So you understand the basis for the anticipation where you're getting earlier onset tumors in subsequent populations?
Yeah, so we're going to publish, there's a paper that's just winding its way through final review. Everybody says final review as if that means it's going to be published, right? It's winding its way through final review. And
Well, if it were cancer discovery, it
but
would be.
I was going to say, I'm happy to share it with you because it may be not in final review. So, so let me put it harshly and then sort of come back, right? Anticipation is an ascertainment bias. And we can show that by looking at the age of onsets in different generations in a family and the nature of the tumor, the type of tumor. When we looked at that, the reason it's an ascertainment bias is that to be a grandparent and to be around so you can give blood, right, for this, you've developed the tumor late, right? To be a parent in the intermediate and to be a kid and see it over 3 generations where anticipation is seen, you have it young. So first of all, it's a setup for an ascertainment bias. But secondly, they get different tumors. The young ones get adrenal cortical carcinomas, and the old ones get either late breast tumors or they get other kinds of tumors that are late brain tumors, things like that. And so it probably has been an ascertainment bias. Everybody's looking for ascertainment. Everyone's looking for anticipation. So that's clearly a statistical hypothesis right at this point, but it led us to the fact that the age of onset is very dictated by the tumor type, which fits the hypothesis.
Okay. Well, thank you very much. A terrific talk, Tom.